Hi. Today we're going to talk about chromosome variation. Chromosome variation is a complicated topic, but I think it's very interesting. And these types of changes can cause things as birth defects, cancer, or can even be drivers of evolution. So I hope you enjoy the lecture, and here comes the voiceover. Here's a normal karyotype of a human male. You can notice that there are many different sized and shaped chromosomes, and the dashed lines indicate the position of the centromere. The position of the centromere is very important in the classification of eukaryotic chromosomes. So if the centromere is in the middle, the chromosome is designated as metacentric. If the centromere is located between the middle and the end, it is submetacentric. If it is located close to the end, it is acrocentric, and the two arms of the chromosome are called P and Q. P is the shorter arm and Q is the longer arm. And if the centromere is located at the end of the chromosome, it's a telecentric chromosome. To visualize chromosomes, we use different staining techniques. We can look for G banding, which stains AT-rich regions. We can look at Q banding, which stains more of the entirety of the chromosome, and this is using quinacridone mustard and visualizing the chromosome under UV light. We can use C banding, which involves the Leishman stain, and this will stain for the centromere, as you can see clearly here. And we can use R banding, which stains for GC-rich regions of the chromosome. Chromosome mutations can occur in a number of ways. You can have rearrangements, aneuploidy, and polyploidy, and I'm going to go through these different scenarios. So chromosome rearrangements involve the rearrangement of genes within a chromosome. And these can involve duplications, which could occur in tandem, which is like right next to each other, or displaced, which involve the movement of this duplication to another region of the same chromosome, or can occur in the reverse order. So I'm going to show you examples of this as well. You can have deletions of genes in the chromosome, and you can have inversions that are both pericentric and paracentric. Aneuploidy involves an increase in the copy number of a particular chromosome. For example, here you see there's a trisomy, so there's 3 copies of one of the chromosomes. So 2n plus 1 equals then 7. So all aneuploidies are an increase or decrease in chromosome number. And this could be, 1 or 2 or 3 chromosomes that are affected, for example. Polyploidy, here we have an autotriploid, which has 3 sets of chromosomes. So instead of being 2n, now the organism is 3n. This is a presence of full extra chromosome sets. So it's not the duplication of individual chromosomes or the deletion, let's say, of individual chromosomes. It is a wholesale duplication of the entire chromosome set. So now I want to look a little more closely at some chromosomal rearrangements. So you have duplications where, let's say here you have E and F, which are duplicated in tandem. And this causes some difficulty in alignment of the chromosomes during prophase 1 of meiosis. So the duplication here has to loop out. But gene duplications don't always occur in tandem. There are 3 types: tandem, displaced, and reverse. So let's say you have a chromosome like predicted that's depicted above, and the gene order is from A through G, and with a red dot refers to the centromere. If you have a tandem duplication of E and F, you have both genes duplicated and then they're in the same orientation. If you have a displaced duplication, then E and F are separated by another gene, or they've moved to another part of the chromosome. Here they've moved to the end of the chromosome. And if you have a reversed duplication, then the orientation of F and E is reversed. And just to be clear, the arrows I'm showing in this slide indicate the orientation of the genes. Drosophila is always a great example of genetics, and here we're going to talk about the bar region. The bar mutation results from a small duplication on the X chromosome that is inherited as an incompletely dominant X-linked trait. This duplication, when you're heterozygous for it, you get a smaller eye, so the number of facets in the eye are reduced. If you're homozygous for the bar mutation, which is a particular type of duplication here in C, you can see that the phenotype gets even stronger. And if you have a chromosome that has a double bar mutation, you can get even a stronger phenotype. So this shows that duplications can have really strong effects on the phenotypes of organisms. What about deletions? Deletions, they also cause a problem during homologous chromosome pairing because you have to loop out the region that has been deleted, which is similar to the effect when you have a duplication, right? Because that region that has been deleted is now no longer homologous with the other chromosome. So A, B, centromere C, D, E, F, G is the normal gene order. And when you delete E and F, you get this. And these deletions can be easily detected using some of the techniques we discussed before for staining chromosomes. Deletions happen in human chromosomes, and the effect depends on the genes located on these chromosomes. You create then a heterozygous chromosome where the deletion of the wild type allele results in the expression of the recessive allele. And this is pseudodominance. Could uncover a phenotype that you would not normally see. There is an imbalance in the gene products then, and some genes need to be present in two copies. Loss of function phenotypes from just having a single copy of a gene is called haploinsufficiency. So you would say that that gene is haploinsufficient. You need both copies of the gene to get a normal phenotype. Inversions are another important type of chromosomal rearrangement. There are two types, pericentric and paracentric. Pericentric inversions include the centromere, where paracentric chromosomes do not include the centromere. For example, here you have your wild type chromosome with gene order AB, centromere, then C through G. Then in a paracentric inversion, you can see here that B has moved to the other side of the centromere, and C and D have also shifted. A paracentric inversion would look something like this, where you have F has changed places with D. So these occur by breaking and rejoining of the chromosome. And there's neither a loss or gain of the number of genes. However, if you break in the middle of a gene, that could be very disastrous to the gene and result in a loss of function. So they have to occur correctly in order for the chromosome to be functional. This changes the position effect gene regulation. So this is where a gene is in the chromosome influences how it is expressed. So the gene could be expressed at inappropriate times or in inappropriate tissues, and this is called position effect. The other aspect of inversions is that they cause reduced recombination. And this has been used in Drosophila to create chromosomes called balancers that inhibit recombination. This is useful if you do not want, let's say, a mutant allele to segregate away from a marker gene that has a visual phenotype. For those of you who are not fly geneticists, you'll probably never encounter a balancer or why it is used, but I just thought I'd put that out there. Paracentric inversions cause an unusual inversion loop during pairing in prophase 1 of meiosis. So you see here we have a normal chromosome at the top that has C, D, and E in the wild type order. And then you're pairing this chromosome with a chromosome that has undergone a paracentric inversion. During prophase 1 of meiosis, the chromosomes form an inversion loop, which allows the homologous sequences to align. These exhibit reduced recombination among genes, and if they recombine, they often produce non-viable gametes. So if we look more carefully at this, we have the inversion loop. If you have a single crossover within the inverted region, and you can see that occurring between, in the highlighted area here, between the genes C and D in the center of the loop. This results in an unusual structure. One of the 4 chromatids now has 2 centromeres. And then one of the chromosomes now lacks a centromere. During anaphase 1, this becomes a problem. As this resolves, the centromeres separate and stretching the dicentric chromatid, which breaks. The chromosome lacking the centromere is lost. And you might see those are the breaks that are caused, the 2 centromeres. And then you have no centromeres in the acentric chromosome. So why might this be a problem? Why would the acentric chromosome not get passed on to the next generation? Well, the centromere is necessary for attachment by the spindles during meiosis. These chromosomes do not get passed to the daughter cells that are produced after anaphase 1. So you get different types of gametes resulting from a single crossover during paracentric inversion. You get the normal non-recombinant gametes, and you also get non-recombinant gametes with paracentric inversions. But these are both viable because you have the full complement of genes. However, the non-viable recombinant gametes, these are lethal because they don't have the full complement of genes that are necessary to form an embryo that's viable. So basically, you need to have E, D, and C on both chromosomes in order to create a viable embryo. Pericentric inversions can also occur. And in this case, we're looking at a pericentric inversion when you compare the human chromosome 4 and chimpanzee chromosome 4. You can see that there has been a pericentric inversion that is around the centromere that causes a difference between human and chimpanzee chromosome 4. Several human chromosomes only differ from those of chimpanzees by a pericentric inversion. Now, these inversions can change gene expression. So it is possible that changes in gene expression in these genes that have undergone a pericentric inversion is responsible for some of the differences between humans and chimpanzees. And chromosome 4 is just an example. There are other chromosomes that differ between humans and chimpanzees in just this way. Pericentric inversions can also be subject to crossing over. So during prophase 1, you get a crossing over with inversion. If crossing over takes place within the inverted region, two of the resulting chromatids have too many copies of some genes and no copies of others. As the homologous chromosomes separate by anaphase 1, you can see that some of the chromatids carry the full complement of all the genes, whereas others either have a duplication, like in the case of A and B, on the lower chromosome, or in some of those chromosomes, they're missing G and F. So the full complement of genes is not in all of the chromatids. So when the sister chromatids separate in anaphase 2, forming 4 gametes, not all of these gametes are viable. So let's look at the gametes resulting from a single crossover and a pericentric inversion. So here are what the parental chromosomes look like during meiosis. You have one normal chromosome that has the full complement of the genes that are necessary, and it has its 2 sister chromatids. And then you have your pericentric inversion chromosome that has an inversion in the order of genes E, D, and C. When you form gametes, you have the parental gametes with no change in the order or the number. You also have a gamete where you have no change in the number, but there is a change in the order of genes, and this is your pericentric inversion. And if you have crossing over, you will have a duplication of F and G, and you lose A and B in one gamete. And then there's a duplication of A and B, and then you're missing gene G and F. So then these are not viable. Translocations can also occur, and this is a kind of interchromosomal change, not an intrachromosomal rearrangement. So you can see here that you have movement of E and F to this other chromosome, which contains MNOPQRS. And you also have QRN moving to the other chromosome. So this is a reciprocal translocation. So translocations involved the movement of genetic material between non-homologous chromosomes. So these are interchromosomal rearrangements. There are 3 types: non-reciprocal, reciprocal, and Robertsonian. If we consider 2 chromosomes, chromosome number 1, AB, CDEFG, and chromosome number 2, MN, OPQRS. So these are the order of the genes on these chromosomes. In a non-reciprocal translocation, you would get chromosome 1 containing A, B, C, D, and G, for example, and then chromosome 2 containing MN, OP, EF, and QRS. So one chromosome is getting more genes than the other. In a reciprocal translocation, you get movement from chromosome 1 to chromosome 2 in a reciprocal fashion. So in chromosome 1, you exchange Q and R with E and F, but you still get the same total number of genes on either chromosome. So there's a reciprocal exchange between the 2 chromosomes in this form of interchromosomal rearrangement, this translocation. So this has important effects on gene expression. there are new linkage relationships which affect gene expression via position effect. If a break occurs within a gene during this process, this can also disrupt the gene's function. So this can be very problematic. So this is a very risky situation. You do not want these translocations to happen normally. Another type of translocation is a Robertsonian translocation. So the long arms of two acentric chromosomes become joined at a common centromere. And then the smaller chromosome often fails to segregate. So you can see here that you produce a new metacentric chromosome and a fragment that is likely to be lost. And this seems to be a driver of evolution. So when we look at human evolution, we can see here that G banding reveals that a Robertsonian translocation in a human ancestor switched the long and short arms of the two acentric chromosomes that are still found in 3 other primates. This translocation created the large metacentric human chromosome 2. And if you look at this figure, the gray lines highlight some, but not all, regions of homology between the chromosomes. So part of human evolution must include this Robertsonian translocation of these chromosomes to form the human chromosome 2 that we have today. Chromosomal variations do not always have positive outcomes, though. And this is the case in, quote, ploidy. And there's different types of ploidy that involve changes in the number of chromosomes. So you have aneuploidy, and the definition of that means a change in the number of chromosomes. You have polyploidy, which is changes in the number of chromosome sets. Aneuploidy was first recognized by Calvin Bridges in 1913. The mechanism of aneuploidy is due to the loss of chromosomes in meiosis or mitosis. And this could be caused by the loss of the centromere due to inversions, which we discussed earlier, or the loss of chromosomes due to translocations, such as a Robertsonian translocation. Or this can occur through nondisjunction in meiosis 1 or meiosis 2. And in the case of cancer cells, these non-disjunction events can also occur in mitosis. Non-disjunction resulting in aneuploidy can occur in a number of ways. You can have non-disjunction in meiosis 1, where the chromosomes are not evenly distributed during anaphase 1 of meiosis 1. In meiosis 2, then you would get gametes that contain certain chromosomes. And in fact, they have 2 copies of that chromosome. And then other gametes that contain none of that chromosome, because one of the chromosomes that was supposed to go to that cell in meiosis 1 didn't get there. So when these gametes are fertilized by a normal gamete that has one haploid copy of the chromosome, you get zygotes that are either trisomic, so that's 2n plus 1, or monosomic, 2n minus 1. If the non-disjunction occurred in meiosis 2, the homologous chromosomes separated properly during anaphase 1 of meiosis 1, but in meiosis 2, you have a disruption in anaphase 2 that leads, for example, in this case, one gamete to get 2 haploid copies, whereas another gamete gets none, and then 2 of those are normal. So when these gametes are fertilized, you get both trisomic and monosomic zygotes, as well as normal diploid zygotes. Non-disjunction can also occur during mitosis. So the improper segregation of chromosomes during mitosis leads for one cell to have only one N, and then another cell that has 2n plus 1. And then these cells go on to proliferate, and you get somatic clones of either monosomic cells or trisomic cells. Such abnormalities can be linked to defects in cell function, such as cancer. So there are different types of aneuploidy. There's nullisomy, which is the loss of both members of the homologous pairs. 2n minus 2. So if 2n equals 46, like it does in the human situation, a nullisomic person has 44 chromosomes. Monosomy, which is a loss of a single chromosome, that is 2n minus 1. So in the case of humans, 2n equals 46. So a monosomic person has 45 chromosomes. Trisomy is a gain of a single chromosome. So that person is 2n plus 1. And if 2n equals 46, a trisomic person has 47 chromosomes. Tetrasomy is the gain of 2 homologous chromosomes. So you have 2n plus 2. So if 2n equals 46, the tetrasomic person has 48 chromosomes. Autosomal aneuploids also occurs in humans. These usually result in embryonic lethality. Therefore, live births of people with these chromosomal anomalies are less common. And this is because there is no mechanism for dosage compensation in autosomes like there is for the X chromosome. The only exception is trisomy 21, which is an extra chromosome 21. This is also called Down syndrome, which was identified by John Langdon Down in 1866. John Langdon Down recognized that these people shared both certain physical characteristics and intellectual disabilities. It was later linked to the fact that there was a duplication in chromosome 21 in these individuals. And that's why it is also called trisomy 21, which is primary Down syndrome. And this was recognized in 1959 by Jerome Lejeune and Patricia Jacobs. The rate of Down syndrome increases with maternal age. And this is caused by the nondisjunction of chromosome 21. When a woman is pregnant at the age of 20, the rate is 1 in 2,000 births. However, by the age of 40, this increases to the rate of 1 in 100 births. By the age of 50, it occurs in 1 in 12 births. Why maternal age is associated with nondisjunction is not known for certain. However, there are theories that suggest that because the second meiotic division takes place immediately before the nuclei of egg and sperm unite to form a zygote, that during the period where eggs are arrested in meiosis 1, the eggs are suspended in the diplotene phase for many years before the ovulation takes place, and components of the spindle and other structures required for chromosome segregation may break down in the long arrest of meiosis. This may lead to more aneuploidy in children born to older mothers, but still this is not known. Polyploidy is a change in the number of chromosome sets. So we have triploids, tetraploids, pentaploids, etc. You can have autopolyploidy, that involve chromosome sets from single species, and allopolyploidy, which is a combination of chromosome sets from 2 species. Autopolyploidy occurs during mitosis occasionally. So you have a 2N diploid cell. It goes through mitosis, and the separation of chromatids is incomplete, and you get nondisjunction, and there is no cytokinesis. So therefore, you produce a 4N cell, which is an autotetraploid cell. Autoploidy can also occur during meiosis. So you start out with a diploid cell, the chromosomes replicate, and you have nondisjunction in meiosis 1. In meiosis 2, you form gametes that are 2N instead of 1N. They are fertilized by normal gametes that are 1N, haploids, and you end up with a triploid zygote. Autopolyploidy leads to problems in meiosis. So for triploids, all 3 chromosomes are homologous. No matter how they align, random segregation will create unbalanced gametes by the end of meiosis 2, and including some gametes that are completely missing the chromosome. So you can get 2N gametes, 1N gametes, and 3N gametes, or gametes that have no chromosomes at all. Alloploidy occurs when you have the hybridization between 2 different species, and this occurs more often in plants. So you have the gamete of species 1 and the gametes of species 2, and these fuse. The organism is functionally haploid and sterile because these different chromosomes cannot pair. They're not homologous, these haploids. But doubling can occur that can result in a functionally diploid, and you can see that here in the F1 generation. So you have a hybrid that is formed. This hybrid produces non-viable gametes for the most part. But if there is a non-disjunction at an early mitotic cell division, you can get the production of an allotetraploid, which has 4N, therefore 12 chromosomes in this case. And then this allotetraploid can produce a full set of viable gametes, here marked A, B, C, G, H, and I. George Karpechenko in 1920 did experiments with cabbage, and he crossed them with radishes. And in this cross, the hybrid was sterile for the most part, but one of the hybrids was able to produce seed after several crosses. And it possessed a 2N, it was diploid, and it had 36 chromosomes. And it amazingly had the roots of cabbage and the leaves of radish. And this is an example of some of the roots that he showed in his paper. Many crop plants are polyploid, and wheat is an example. Wheat is made of the chromosomes from 3 different species. And why is this? Genome size is directly proportional to physical size, and cultivated plants are bigger. So in the parental generation, we had presumably a cross between the einkorn wheat and wild grass. And that produced 2 different types of gametes, which formed a hybrid. And because the chromosomes were homologous enough that they could actually pair. A mitotic non-disjunction event led to the production of Elmer wheat, which is 4N and it has 28 chromosomes. So you have a duplication of the entire chromosome set. This was crossed to wild grass again, producing another hybrid that was 3N. Through mitotic non-disjunction, we eventually ended up with the bread wheat, which we use today, and its genome was 6N, and it had 42 chromosomes. And this is known as a hexaploid because it is 6N. There are other examples of polyploidy in crop plants. Potatoes are 4N, banana is 3N, peanut is 4N, and sweet potato is 6N. And these all contain multiple copies of chromosomes from the same species. Where tobacco, cotton, wheat, oats, sugarcane, and strawberries are Allopolyploids. So they contain chromosome sets from different species that were hybridized, and this created the ploidy and also the traits that we selected for in these crop plants. But polyploidy is not always a good thing. Changing the number of chromosomes in a cell can also cause cancer. So a normal human cell that is healthy has 46 chromosomes. But for example, here, this colorectal cancer cell has 59 chromosomes. And it is both polyploid and has a number of chromosomal rearrangements. So it has deletions, inversions, translocations. Tumors appear to be associated with these chromosomal abnormalities. For example, myeloid leukemia is caused by a reciprocal translocation, resulting in the disease, which is a leukemia of bone marrow cells. The fusion protein that is caused by the translocation fuses the BCR gene with c-Abl, and this stimulates increased unregulated cell division. And so what is leukemia? It is a cancer that starts in blood-forming tissue such as bone marrow and causes large numbers of blood-related cells to be produced and enter the bloodstream. And this, of course, can be very, very dangerous and can lead to death. So here are some genetic terms and concepts that I'd like you to remember from this lecture. Chromosome rearrangement is a change from the wild type in the structure of one or more chromosomes. Pseudodominance, expression of a normally recessive allele owing to a deletion on the homologous chromosome. Haploinsufficient gene. This gene must be present in 2 copies for normal function. If 1 copy of the gene is missing, a mutant phenotype is produced. Paracentric inversion, a chromosome inversion that does not include the centromere in the inverted region. Pericentric inversion, a chromosome inversion that includes the centromere in the inverted region. Position effect, dependence of the expression of a gene on the gene's location in the genome. Translocation, movement of a chromosome segment to a non-homologous chromosome or to a region within the same chromosome. Aneuploidy, change from the wild type in the number of chromosomes, most often an increase or decrease of 1 or 2 chromosomes. Polyploidy, possession of more than 2 haploid sets of chromosomes. I'm leaving you with an image of the dengue virus. Next time we will discuss bacterial and viral genetic systems. See you next time.